We are done with what? Monohybrid cross. So the next one is what? Dihybrid cross. Dihybrid what? Cross or dihybrid inheritance. Dihybrid in what? Inheritance. This is evolve or evolving, crossing of two genetic character. The hybrid inheritance is evolving the cross of two genetics what? Character. Monohybrid evolve what? The one generic what? Character. But here is evolving the what? The cross between two living organisms of two generic what? Character. So, and here the first one is what? The first filial generation of the hybrid cross can be obtained by cross diagram. That is, the offspring, the what? The F1 generation can be what? Obtained by cross diagram. Can be obtained by what? By cross diagram. While the second filial generation, the what? The second what? Filial generation can be obtained by punnett square. Punnett what? Square. And the total number of offspring. So what? Total number of what? Offspring can be found. Sorry, the total number of offspring in the hybrid inheritance is 16, which is what? N square. Where the n is what? Is 4. 4 square is what? 16. And the phenotypic ratio, typic ratio for the hybrid cross or the hybrid inheritance is 9:3:1. Where what? The phenotypic ratio is 1:2:4:2:2:1. There is a short method or formula that we can use to what? To determine the genetic ratio of what? Of the hybrid inheritance. This is what? The hybrid, and this the hybrid is what? It is in accordance with Mendel's second law of genetics. of what? Genetics, which is what? Law of what? Independent assortment of what? Of gene. So let us solve one question on how to what? To apply this dihybrid what? Course. Let us solve this question for the dihybrid. We are given a certain plant of two allele for a flower, red and white, and two allele for plants, height, which is what? Short and tall. We can see that what? This question now is deal with what? Two cause of two genetics what? Factors. Here we have what? Talking of what? Red and white flower. Is it that with what? With short and what? Tall. In the first cause, which is monohybrid, that one is deal with what? One. And the one we have there is what? It just deal with a black rat and a white rat. Here we are talking of what? Red flower and white flower. And short plant and a tall plant. Do with what? The cousin of what? Of two traits, which is character. We have here, which is the hybrid. We are now say what? Solution. What? Solution. Say what? Let the what? Genotype for we have what? Red flower and white flower, tall and short. For tall and what? Red flower, tall and what? Red flower plant. Their genotype. Let is equal to what? For tall, we all know that what? Tall is, yes, tall is what? T, T. Red is what? This. And again, let what? Genotype for short and white flower plant should be what? Let that all be what? This and R. But I said our first filler generation, we can obtain it by what? Cross diagram. So if the parent here, if the parent here is what? The two parents we have there are this, and we have T, T, R, R. And here we have what? Small t, and what? Small r, small r. So if meiosis occur, meiosis, this will split into this and this. This will split into two through the process of what? Meiosis. And here now, we will have what? TR. Here we have TR. So at this side too, we have TR, we have TR. when the crossing, and this place is what? This is what we call what? What? Gamete. So if the crossing now occur with this, we have it with what? So what do we have here? We have what? T, small t, r, small r, and here too we have what? T, small t, r, small r. The same thing we have in others too. So this same thing we have there, we have this with what? With this. Then this one again with this. So we have what? A photocopy. When this combine here, small t, small r, small t, small r, small t, small r, small t, small r. You have what? T, r. You can now see that what? This is the what? Fertilization, and everything you have here is F1 generation. So the next thing that we'll do now is what? We will now take what? Two parents here in F1 generation, take it to the what? F2 what? Generation. For the second filial generation, we will apply what we call what? Punnett what? Square. So by using the Punnett square, we will have stuff like this. Four gametes. This is what we call what? Here we have what? Gamete. So the first one, if you have what? T, capital R. Let me use this thing for it. Uh-huh. So from here now, four types of gametes are what? Are formed there. The first one is what? We can have what? T, capital R. The second one, we have what? T, small r. The third one, we have what? Small t, capital R. Here we have small t and small what? Small r. And here, we have the same thing, which is what? Small t, capital T, small r, small r, small t, capital R, small t, small r. So let us look at what? The breeding. For this now, we will now try what? T times T will give us what? 4. It will give us what? 4. This R times R. Let me use this to be clear. T, let me use to be for the convenience sake. This T is this, What? And what this R is what? Is this. So when this T come in, we will have what? T and we have what? Second R. Multiply it like this. Abi? And likewise, this one too. We have what? T. No, this T up first. T and R. We have it in this form. That will be T small r. So here again, multiply this by this, small t r, we have what? Capital T, capital R. So here we have what? Capital T, we have capital what? R, which is what? And here we have what? No. We have small t and we have what? Small r. So if you multiply this again, we have what? T and we have what? R. So if you multiply it for this, we have T small r. If you do that again, capital T, capital R, and this side, we have what? This T is this. This the mistake. This T is this. This T is what we have a small r for this, and we have what? Another what? Small r. So capital T, small r, then we have small t, big R. T, R, we have capital T, small r. And here, what do we have? T and capital R. So for this one, small t and capital R. Then for this one, small t, capital R. Here we have what? Capital T. We have small r. Here we have small t, big R. So here we have what? Small t, big R. We have big what? Big R. Here we have what? Small t, small t. We have small t, small t, small r. So here we have what? Small t, capital what? R. And the next one here is we're having what? Capital T, capital R. So we have small t, small r. Here we have small t, small r. Up we have capital what? Capital T, small r. Here we have small t, small r. Here we have small t, small r. We have small t, small what? Small r. So here we have what? Small t, small r. If you look at all this now, the product we have here, let us check it. We are having what? How many homozygous tone and homozygous thread? homozygous tall and homozygous thread. We have what? Result T T R R are what? Called homo what? Homozygous tall and what? And red. How many? How many of them are what? We have just what? One. Which is what? One. Well, again, this. Let me go for this one. How many are what? Homozygous what? Are homozygous tall that have what? T T and have big R, small r. The meaning of this is what? They are what? How many of them are homozygous got tall and heterozygote red? Homozygous tall and homozygous what? Red. Yeah, let us count it. One. Homozygous tall, heterozygote red. We have two here. Homozygous tall. That is, we have one and what? And two. Two. So answer here is what? Is two. So the next one is what? We are done with this. Let me put here. We are done with this. How many is there is what? C small and R. So among these now, so T this is what? Homozygous tall and homozygous red. Count it here is two. Homozygous heterozygous tall, heterozygous what? Red. There are four. Then heterozygous tall, homozygous red, homozygous white. They are what? One. No, this one is two. This is the second one. So here we have what? Two is two. And we have homozygous what? Short and homozygous red. This one is homozygous short and homozygous what? Red. How many? How many? Short and homozygous red. This one is what? Is one. So after this, we have homozygous short and heterozygous red. Homozygous short and heterozygous red is not there. Homozygous short and heterozygote zygote red. How many? Homo short, heterozygote red. How many? One, two. We have one, two. This place is two. What the next one? What the next one? We have look at this. Homo tall, heterozygote red. You have done it. this out of homo what? Short. Heterozygote what? Short. Short will be heterozygote short. We have this, and this, which is what? Homo short, and homo zygote red. That will be what? How many be that? 1. So altogether, 1, 2, 3. So that will be what? 5, 10, 10, 15 is what? 16. Yes. What are we determining now? Is genotypic what? Ratio and phenotypic what? Ratio. So by rearranging this now, the genotype here, the genotype or genotypic what? Ratio from this expression, genotypic what? Ratio, it will now be what? We have 1, 2, 1 ratio 2. I'm supposed to put this one here. It will now be what? 1 ratio another what? 2. Then ratio 4. Then ratio this 2. Ratio this 1. Ratio this what? 2. Ratio this 1. This is what? Genetic ratio. This my homozygote tall and homozygote white is supposed to be in this side. So you now have it in this form. And the next thing is what? If you look at it now, this is what? Here. 4 plus 2 plus 1, 7 plus this 4 plus 6, 7, 8, 9. There's a lot of mistake here. We want to add 16. Add it together 4 plus this one making 6 plus this one making 10 plus 3 here making what? Supposed to have 16. Yes, 9, 3, 3, 1. Yes. So we have this together 2, 2, 2 plus this. Let me say this 3. Yes, this is what? Genetic ratio. But for what? For phenotypic ratio. You know what? Typic what? Ratio here. It should be what? 9 ratio 3, 3, 1. 9 what? Ratio what? 9 ratio 3, 3. Ratio what? 1. So from this sketch, we have 9 ratio 3, 3, 1 as our what? Phenotype. So that is all about what? Dihybrid course. But there is a particular shortcut that I would like to what? To insert. If your exam is multiple what? Choice. Let me insert the shortcut here. So it will be very useful for us. The shortcut is if you are asked, you know, I said the course, yes now, I said course can be divided into 3 parts. The course or abbreviation, we have what we call what? Mono what? Monohybrid. Monohybrid what? Course. We have what we call di what? Dihybrid. Serious. Dihybrid cause, and we have what we call tri what? Trihybrid what? Cause. This monohybrid deal with what? One character. Di, two character. Tri, four character. But you can ask that what is the what? Phenotypic ratio. You know what? Formula to tell me what? V E know what? Typical what? Ratio. So the formula used to calculate what? Phenotypic ratio of any hybrid cause is 3:1 exponential what? N. This is the general formula that we can use to find the what? The genotypical what? Phenotypical ratio either for dihybrid, monohybrid, or what? Trihybrid. To know the phenotypic ratio now, for monohybrid, our N here is what? 1. So for what? For mono, I what? Hybrid, hybrid, N is what? N is 1. So what will be the what? This one is 3:1 exponential 1. We have what? 3:1. We have done it. But if we have dihybrid, for what? I what? Hybrid, our N equal to what? 2. What is the phenotypic ratio? It will be what? 3:1 exponential what? 2. You now have what? 3:1 into what? 3 into 3 what? Ratio what? Ratio 1. We will multiply everything you have here by this. And let us do it, and we check the what? The answer. This 3 times 3 here to give us what? 9. This 3 times 1 to give us what? 3. Then this one times 3 to give us what? 3. This one times 1 to give us what? 1. This is what? The phenotypic ratio for what? For dihybrid. So how are we going to know the what? The phenotypic ratio for trihybrid. For what? For trihybrid, cause our N equal to the what? 3. So here you now have what? 3:1 into the what? Raised to power 3. Here we have what? 3:1 ratio 3:1 and ratio 3:1. Because you write it what? In 3 times. But as we all know that what? These two already, they are give us what? 3:what? 9:3 ratio what? 3:1. Abi, you now multiply everything you have there with this one now. 3 times 9, we have 20 what? 27. Ratio 3 times 3, we have 9. Ratio 3 times 3, we have another 9. Abi, ratio 3 times what? Times 1, we have 3. Then multiply everything again by what? By 1. 1 times 9, ratio what? 9. 1 times 3, ratio what? 3. 1 times 9. Where am I now? Yes, 1 times 9, 9. 1 times 3, 1 times 3, second 3. 1 times 1. So we can now rearrange it to have it in this form. 2, 7, ratio 9, ratio 9, ratio 9, ratio 3, ratio 3, ratio 3, ratio 1. So this is what? The phenotypic ratio for what? For trihybrid. So that is what the short formula. But what is the short formula for us to know the what? Genetic what? What is the short formula for us to know the what? The phenotypic ratio. Yeah, the formula for genotypic ratio, pick what? Ratio is 1:2 exponential what? N. So for this one now, yeah, let us look at what for monohybrid. For mono, I what? Hybrid. So our monohybrid N is what? Is 1. Yeah, let us put it here. We have what? 1:2:2:1. It will later give us what? 1:1:1:1:1. Yeah, for di what? Dihybrid, N equal to what? 2. So for N equal to 2 now, here we have what? 1:2:2 square. And if this is the case, you can rewrite this to be in this form as what? 1:2:2 into bracket what? 1:2:2:2. You write it twice. If you write it twice, we now what? Multiply everything you have what? In fact, let us multiply and check what are we going to have. Yeah, 1 times 1 here, we have 1. 1 times 2, 2. 1 times 1, 1. Take 2. 2 times 1, 2. 2 times 2, 4. 2 times 1, 2. The next one, 1 times 1, ratio 1. 1 times 2, ratio 2. 1 times 1, ratio 1. If you check it here, it will be almost the same. See it here. Ratio 1, 1:2, 1:2 again. We have 4, we have 2, we have 1, we have 2, and we have what? Ratio what? Ratio 1. So we can try what? For try what? Try hybrid. Try I what? Hybrid. Find the what? The genetic ratio for what? Try hybrid. So that is all about what? Dihybrid and a what? And a monohybrid. So the next aspect of genetics is we have some condition that they disobey or they deviate out of what? Out of Mendel's law. Both first law and the second law. What are the conditions that deviate from the what? From the Mendel's law? We have what we call what? Codominance. We have incomplete dominance. And so on and so forth. So in my next video, I'm going to what? To move to the what? What are the conditions that disobey or the deviate out of what? Of Mendel's law of genetics. Thanks for watching.